My name's Chrinstine Andersen. I'm a junior, rising senior. I work in the Thompson lab, and the Thompson lab is interested in the different mechanisms behind social behavior and what molecules are responsible for regulating social behavior and how they work in the brain. My specific peptide that I'm looking at is called Vasotocin, and it is involved in all sorts of behaviors such as courtship and aggression. Specifically, what we're looking at is mapping out the receptor and where in the brain this receptor lies in Goldfish. I'm looking at how this peptide can affect behaviors differently in different settings. For example, it could promote an approach response towards another animal in one setting, whereas it could promote withdrawal from another animal in a different setting. I'm going to be looking at courtship because we know Vasotocin in a male-to-male interaction cause males to withdraw from other males, but we're wondering if it's going to promote males approaching females in the courtship setting, which would be really interesting if the same peptide is able to create opposite effects based on two completely different external stimuli. So far I've been working on mapping out where the receptor is in the brain. Immunohistochemistry, which is what we're using to characterize these receptors, is something that you use where you slice a brain up into very thin sections so you can look at individual cells, and then you take advantage of the immune system and other animals' ability to produce antibodies towards a specific receptor, in this case the Vasotocin receptor. Then you conjugate another antibody to that antibody. So it's a goat anti-rabbit antibody, so my anti-Vasotocin was raised in rabbit, so we're now trying to tag for anything that's rabbit on the fish brain slides. It's also conjugated to a fluorophore, so it's wrapped in foil because it's very light sensitive, because that's what makes it fluoresce on the microscope. And you can indirectly visualize the receptor through a fluorescent microscope. After we map the receptors, I'm going to look more specifically at how they work. So we think that these receptors dimerize in the membrane, and what actually exists in the membrane is a dimer of the receptor itself and a truncated version of the receptor. I think that the way that these Vasotocin receptors dimerize might possibly be influenced by external and internal stimuli. So I'm going to specifically look if exposure to a pheromone will change the way that the receptors are able to dimerize, which would potentially lead to a functional difference in the receptor.